Welcome to another episode of The Future of Product. Today, we're diving into a story of a major tectonic shift in biomedicine, the incredible success of C2S-Scale. This is a massive 27 billion parameter AI model that has successfully taught itself the fundamental language of human biology. We'll start by unpacking the ingenious trick, the cell-to-sentence framework, that let this language model master complex genomics data. This unlocked unprecedented reasoning, from predicting cell types all the way to genuine scientific invention. We'll trace its journey to a tangible, life-saving discovery, identifying silmutisert as a novel cancer therapeutic. This isn't just a powerful model, it's a new blueprint for accelerating discovery itself. Before we dive in, please take a moment right now, hit that subscribe button so you don't miss out on any of our future breakthroughs. Now let's dive in.
Welcome back. Today we are opening up a massive research initiative, something that really feels like it shifts the tectonic plates in biomedicine.
Yeah, it's pretty stuff.
It is. It's the story of how a 27 billion parameter language model, AI on the scale we talk about for general intelligence systems, didn't just, analyze biological data.
No, it went much further.
It actually made a validated, potentially life-saving scientific discovery right in the thick of the fight against cancer.
That's exactly right. We're diving into really a core challenge in cancer immunotherapy, which is how do you make these so-called cold tumors visible?
The ones that hide from the immune system.
Precisely. They're the aggressive ones. They have these sophisticated ways to evade detection. They suppress the surface antigens.
Those are the molecular flags, right, that the T cells need to see.
Exactly. Without those flags, the T cells just fly blind. So we desperately need a way to force those cancer cells to, well, essentially paint targets on themselves.
And this AI we're discussing today, C2S-Scale, it found a way.
It generated a precise, testable, and ultimately successful method to do exactly that.
Okay, but the core of this breakthrough, it wasn't a new kind of neural network, was it?
No, not really. It was more about a radical data engineering trick, actually.
Right. So our mission today is to unpack how Google and Yale trained this model, C2S-Scale, which, by the way, is based on the Gemma 2 architecture, known for being highly scalable, how they trained it to speak the fundamental language of human biology.
It's really a paradigm shift, in how we approach these foundation models for single-cell biology.
So instead of complex, custom-built models...
Yeah, instead of those bespoke architectures, the cell-to-sentence framework C2S, they call it, it translates this really high-dimensional single-cell RNA sequencing data.
Okay, cRNA-seq. That's the full gene expression profile for one cell.
That's the one. It translates that complex profile into simple, ordered text.
Text. Okay, let's unpack that. We're talking about taking a cell's numerical genetic readout.
Yeah, thousands of numbers.
And turning it into a sequence of gene names. A cell sentence.
That's the concept.
So we need to understand how this... Well, it sounds like a massive simplification, doesn't it? How does it keep enough information to actually power a huge LLM?
That's the key question, right?
And what do the scaling laws look like when you apply them to biology? Does it work the same way?
We'll get into that. The scaling is really interesting.
And most importantly, how did this whole framework lead directly to validating this drug, silmutisert or CX-4945, as a new kind of immune amplifier?
Indeed. We'll cover the C2S framework itself, its foundation. Then the dramatic effects of scaling up to 27 billion parameters.
Okay.
Its ability to reason about, complex multicellular contexts. And then, the specific virtual screen, the dual context one, that led to this breakthrough.
Okay, let's start with why. Why do we even need C2S-Scale? We already have several specialized single-cell models out there, foundation models or SCFMs.
Right, things like SCGPT, Geniformer.
Yeah. Those are designed specifically for this complex biological data, these vectors. So what was the fundamental problem with those? What limitation did C2S-Scale need to get around?
Well, the core limitation was really architectural constraint. These specialized SCFMs, they're custom built, right, to deal with the numerical complexity of the data, these high-dimensional vectors with thousands of gene expression values.
Right.
But because their architecture is so specialized, they bump up against two major sort of interconnected problems you see a lot with bespoke AI systems.
So they built their own specialized engine, but maybe it couldn't use the same kind of high-octane fuel that the general AI models use.
That's a fantastic analogy.
Yeah.
Problem one is scalability. We know from general LLMs that performance scales dramatically, often, exponentially, once you cross that billion parameter threshold.
Right. Bigger is often better.
But these custom architectures, they're inherently limited in how large and, crucially, how efficiently they can scale. They can't just plug into the highly optimized training infrastructure built for models like Gemma or Llama.
And if you can't scale up like that, you can't really access that higher level of reasoning, that sort of emergent capability we now kind of expect from big AI models.
Exactly right. Which leads to problem two, flexibility.
Mm-hmm.
Because these models speak this very technical language of gene vectors and numerical matrices.
Mm-hmm.
Integrating other kinds of relevant information gets really clunky. Let's say you want to feed in natural language text, maybe from a paper abstract or a clinical note or even a database of molecular interactions.
Stuff that's not just numbers.
Right. The model needs these complicated custom interfaces just to translate that text into its own numerical world. It really hinders the seamless integration of different knowledge types, different data modalities.
Okay. So the Google and Yale team basically said, let's not fight the architecture. They sidestepped those constraints by focusing like a laser on the input data.
Exactly. They engineered the biological data itself to perfectly fit what a large general-purpose LLM naturally prefers to eat, text. And that's the cell-to-sentence framework, C2S. That is the entire premise, yeah. And the brilliance of it, really. It took the fundamental unit, the cRNA-seq profile, which, can have data for 20,000 genes or more, and convert it into this textual sequence, a cell sentence.
I think we need to really visualize this. It sounds like you're throwing away a ton of information. How does that numerical vector, all those numbers, become just a list of words?
Well, the mechanism is surprisingly simple. It relies on a core biological fact.
Yeah.
Not all genes are expressed equally in a cell. Okay, some are working harder than others.
Exactly. So for any given cell, you take those thousands of genes and you systematically rank order them, based purely on their expression level, most expressed down to least expressed.
Right.
Then the C2S framework just selects the names of the top K most highly expressed genes.
Mm-hmm.
Typically, K is around, say, 200 to 500 genes.
Okay, a fraction of the total.
A fraction, yeah. And it strings those gene names together in that rank order.
So if MEL-OT1 is the most active gene in a cell, it becomes the first word in the sentence. Correct.
Yeah.
And if TMSB4X is the second most active, it's the second word. So the sentence reads something like MELOE-TAN1, TMSB4X, BTAM, EEF1A1. Just listing the top gene names in order. That's exactly it.
Yeah.
And the LLM reads that sequence, just like it would read a sentence in English or any other language. The gene names are the tokens, the words.
And the order tells it something.
The rank order establishes the linguistic relationship. You could think of it like a grammatical structure, telling the model about the relative abundance. It's essentially teaching the LLM the alphabet and the basic grammar of how a cell expresses itself.
Okay, let me push back on this a bit, just playing devil's advocate as a biologist might. If we're only using the rank order and we're ignoring the actual numerical magnitude, like the difference between a gene expressed at 100 counts versus one at 1,000 counts, even if they're both in the top 5, aren't we throwing away potentially crucial quantitative information?
That's a fair challenge.
I mean, how do we know that a 15% difference in how much a gene is expressed isn't the critical signal that decides if a cell becomes cancerous or not?
That is absolutely the central question they had to address. They knew if this conversion meant massive information loss, the whole LLM approach would just fall flat.
Right.
However, what they demonstrated was that in log space, which is how biologists typically analyze gene expression anyway, to handle the wide dynamic range.
Okay, log transformation, standard practice.
Yeah, in log space, there's actually a very strong linear relationship between a gene's rank in the cell sentence and its original normalized expression value.
A linear relationship. So the rank alone tells you a lot about the actual value.
Quite a lot, yes. And they proved this with a reversibility test.
Okay, what did that involve?
They trained a very simple linear model. Its only input was the gene's rank from the cell sentence, and its task was to predict the original numerical expression value.
And how well did it do?
The result was an R-squared value of 0.85.
0.85. Okay, for listeners, R-squared tells you how much of the variance in the original data is explained by the model. So 0.85 means?
It means that 85% of the total variance present in the original complex numerical data is retained simply by knowing the rank order of the genes.
Wow. Okay, that's actually really compelling. It suggests that while, yes, you lose maybe 15% of the precise quantitative detail,
Mm-hmm.
the crucial signal, the relative importance, the prevalence of different gene programs running inside the cell that's largely preserved, the LLM doesn't need to know the cell is screaming a gene name, just that it's expressing it louder than 99% of the others.
Precisely. And this conversion, this textual representation, it yields these key benefits we talked about.
Yeah.
The first, obviously, is scalability.
Right, because it's just text now.
Yeah. By using text, they could instantly plug into existing, highly optimized LLM infrastructure like the Gemma 227B architecture.
Yeah.
No need to design custom compute graphs or memory management for weird biological data formats. They got all that massive engineering effort essentially for free.
Makes sense. And the second benefit you mentioned earlier was prior knowledge integration. This feels like where the 27 billion parameters, already trained on tons of human language, really start to pay off in biology.
Absolutely. Think about it. When a custom SCFM sees a gene represented just as, say, index number 4721 in a vector, yeah, it's just a number, meaningless on its own.
Right. But when C2S-Scale sees the token, malate1, the LLM instantly accesses all the vast knowledge it picked up during its pre-training on billions of text tokens.
From reading scientific papers, websites.
Exactly. Scientific literature, abstracts, general biological texts on the web. The model already knows things about malate1, maybe that it's a long, non-coding RNA, often linked to metastasis, or involved in nuclear functions even before it analyzes the specific expression data in front of it.
So wow, they didn't just teach the model the grammar of the cell sentence, they implicitly infused it with the entire scientific history associated with the words in that sentence.
That's a great way to put it. The model comes preloaded with millions of context clues about what genes do, how they interact, just because it read about them in papers.
Precisely. It transforms the learning process. It's not just learning correlations from numerical data anymore. It's learning sophisticated, text-supported biological relationships. Okay, so once they validated this language, this cell sentence approach, the path was clear. Make it bigger. Scale it up. Let's talk about the sheer scope of the training here. We're talking models from 410 million parameters up to 27 billion. Yeah, a whole family of models.
But the data corpus itself is just enormous. Tell us about these 50 million transcriptomes that resulted in over 1 billion tokens.
The scale is pretty staggering. They pulled together over 50 million individual transcriptomes from both humans and mice.
Where did all that data come from?
Mainly from major public repositories and atlases, CellXGene, the Human Cell Atlas, places like that. This data covered everything: healthy cells, disease cells, cells from loads of different tissues, different developmental stages.
A huge variety. It's huge. And all this massive high-dimensional molecular data, plus the associated metadata like tissue type or disease state, and any descriptive text they could find, all of that got converted into this corpus of over 1 billion tokens of cell sentences.
Okay, let's pause there for a second. If you think about the bigger picture, just the data engineering involved.
Yeah.
Harmonizing 50 million single-cell profiles, which are notoriously messy, right? They're captured using dozens of different lab protocols in thousands of different labs around the world.
Yeah, it's a nightmare usually.
Just cleaning that up and getting it into a consistent format for an LLM, that's a monumental achievement in itself. It kind of suggests that these global biobanks, the data quality and accessibility, are finally reaching a point where this kind of massive AI training is actually feasible.
That's a really crucial point. The sheer effort involved in cleaning, normalizing, organizing that molecular chaos into a coherent training set. It's a testament to the infrastructure and the curation work being done by those biobanks.
Absolutely. Without that foundational curated data, a project like this just doesn't happen. The AI is only ever as good as the reality you feed it.
Right. Garbage in, garbage out, even at 27 billion parameters. Okay, so the fundamental hypothesis here rests on those established scaling laws we see in general LLMs. The idea that bigger models trained on more data just perform better, often dramatically better. Did those laws, the ones that govern things like poetry generation or coding, actually hold true when applied to this fundamental language of biology?
They absolutely did. And that's maybe the single most critical validation of the whole C2S framework.
Yeah, it followed the same curve.
Yeah. The results clearly show that performance consistently improved across all the different tasks. They benchmarked things like tissue classification, cell type annotation, even generating new cell profiles conditionally.
Okay.
Performance went up steadily as the model parameter count increased, right up to the 27 billion parameter version. And it wasn't just a gentle improvement. It was that sharp, predictable climb you see in general NLP scaling.
Wow, so that really does prove that this language of biology, encoded as text, follows the same kind of statistical regularities as human language. The bigger model can just capture more complex, more nested biological relationships.
Exactly. It's like understanding subtle sarcasm or complex metaphors in human language. That capacity to internalize billions of gene function relationships seems to only emerge at that massive scale.
Okay, so let's look at the breadth of tasks. How did C2S-Scale actually stack up against those smaller, specialized SCFMs, SCGPT, Genoformer, on the sort of traditional hard biology tasks?
Well, on those standard tasks, it performed very competitively, sometimes slightly better.
You have an example?
Yeah, for instance, cell type annotation. They used a diverse immune tissue data set. C2S-Scale achieved 95.43% accuracy in predicting the cell type, and it did this in natural language, just outputting the name like T-cell or macrophage.
Okay, 95.43%. How did the others do?
Slightly lower. SCGPT got 93.1%, Genoformer 94.0%. So it showed that this text-based model could handle the traditional numerical classification jobs just as well as, or even a bit better than, the dedicated numerical models.
But presumably where it really shines, where it pulls ahead, is on tasks that leverage its language core, the things that were difficult or impossible for the specialized models.
Exactly, the unique sort of multimodal capabilities.
What stands out to you as the most powerful example of that unique versatility?
For me, it's the integration task-matching single-cell data to bulk RNA-seq data. That was extraordinary.
Okay, remind us bulk RNA-seq is different.
Yeah, bulk RNA-seq gives you an average profile from thousands of cells in a tissue sample. It's not single-cell resolution. C2S-Scale was asked to match individual cell sentences to the corresponding bulk profile from the same tissue. This is super important for integrating different types of clinical data.
Okay, but here's the kicker. C2S-Scale had never been explicitly trained on bulk RNA-seq data, never seen it before. So the model had to figure out, basically on its own, how to, sum up the language of individual cells to match the language of the whole crowd.
Precisely. It had to learn a biologically meaningful representation that actually held true across different scales of biological measurement, from the single cell up to the tissue level.
That implies a really deep understanding.
It does. It's like if a cell sentence is a word, the model learned how to synthesize those words into a coherent paragraph, the bulk profile, without ever being explicitly taught paragraph structure. It showed real generalization from micro to macro expression states.
Okay, that's impressive. And finally, what about explaining itself? When you ask it to interpret its findings, talk about the biology in plain English. How did C2S-Scale do compared to the big general-purpose LLMs like GPT-4o or Gemini? They're great at summarizing text, but can they handle the nuances of gene expression?
Well, when the subject is specifically biology, C2S-Scale came out on top. They used a benchmark called CQA, single-cell question answering.
Okay.
The model gets a cell sentence and has to answer a question about the cell state or function in natural language. C2S-Scale outperformed even the most advanced general LLMs on this. It beat GPT-4o by about 3% in BERT score.
Okay, BERT score. We should quickly explain that. It's not just about word overlap, is it?
No, exactly. BERT score is a metric that looks at semantic similarity. It uses contextual embeddings, deep learning features, to judge if the generated answer means the same thing, functionally or biologically, as the gold standard reference answer.
So a 3% advantage in BERT score means C2S-Scale's explanations weren't just fluent and plausible.
Right.
They were significantly more aligned with the actual biological truth represented in the reference answers compared to what a top-tier general LLM could produce.
Yeah. It really confirms that specializing a big LLM with massive amounts of domain-specific context, even using this simple text conversion trick, makes it a much, much better scientific partner than even the largest general models when you're talking about that specific domain.
Okay, so we've established C2S-Scale can classify a cell, answer questions about it, but real biology, it doesn't happen one cell at a time, right? It's all about context.
Absolutely. It's about communities of cells, tissues, responses to the environment.
So true discovery means reasoning across multiple cells, maybe different conditions. How did C2S-Scale use its, massive context window and its natural language abilities to do this more advanced reasoning?
Well, the key enabler here is the LLM architecture's inherent ability to handle really long sequences of tokens. In this case, the Gemma architecture could handle up to 8,192 tokens.
That's a lot of cell sentences.
It is. It allows you to feed not just one cell sentence, but dozens, maybe even hundreds of related cell sentences into a single prompt, a single input. And this multi-cell context lets the model start interpreting biology at 3 distinct scales.
Okay, so we go beyond the single cell level, which is like basic annotation.
Right, standard stuff. And we move up to the cluster level. Tell us about this novel task they defined. Cluster captioning. That sounds interesting.
It is pretty neat. So cluster captioning is an interpretation task. You know how biologists often group cells with similar gene expression profiles into clusters on a plot?
Yeah, standard analysis step. You get these blobs of dots.
Exactly. But figuring out what a cluster actually means biologically usually requires a human expert. They'd have to look up the top genes expressed in that cluster, go read about them, and then manually synthesize some kind of explanation. These look like activated T cells because they express genes X, Y, and Z.
Okay, so C2S-Scale automates that synthesis.
That's the idea. The model was fed all the cell sentences belonging to a specific cluster, and its job was to generate a concise, biologically meaningful description, a caption for that group of cells.
And how did it do? It significantly outperformed baseline LLMs on this task, again using BioBird score to measure the biological accuracy of the generated caption. It really confirmed the model's ability to synthesize these complex high-dimensional patterns into coherent functional summaries.
I can see how that would be useful. For a working biologist, that speeds up the interpretation phase of an experiment dramatically.
Massively, yeah.
Okay, and if you take that one step further, scale it up again, you get to the dataset interpretation task, where the model basically writes the abstract for an entire study.
Pretty much. Yeah, the dataset interpretation task demands the highest level of abstraction. The model might receive hundreds, even thousands of representative cell sentences from a whole dataset.
Wow.
And its task is to generate a high-level summary, like an abstract, describing the key biological features of the entire dataset. The main cell types present, the tissue it likely came from, any disease or treatment condition reflected in the data.
And could it generalize? Could it do this for data it hadn't seen?
Remarkably well, yes. Even for completely novel, unseen datasets. This really moves the AI beyond just being a data processor, becomes more like a scientific narrator, capable of distilling these vast quantities of molecular detail into a coherent story, a research narrative.
That really is advanced reasoning. Okay, let's pivot slightly. Spatial reasoning. This is notoriously hard in biology, right?
Oh yeah, very tough.
Trying to predict where a cell physically sits inside a tissue based only on its gene expression. It feels like trying to guess where someone lives based only on the clothes they're wearing. You need more context.
That's a good analogy. You need neighborhood context.
So how did C2S-Scale try to infer these spatial relationships using just the transcriptomic data, the cell sentences?
Well, they use that multi-cell context window again. They essentially treated spatial organization as a sequence modeling problem.
How?
In the training data for experiments where they did have spatial location information, they would sample cells known to be physically close to each other, say, all cells within a 20-micrometer radius in a slice of litter tissue.
Okay, true neighbors.
Right. And they'd encode the cell sentences for all those neighboring cells into the same long context prompt. So the model implicitly learns that these specific cell sentences appearing together in one input are somehow related. Spatially related.
Okay, it learns proximity correlation. But as you said, spatial relationships are often driven by specific molecular interactions. Like, a T cell needs a specific receptor to talk to a specific ligand on a cancer cell.
Direct communication.
Did they just rely on the model figuring that out from the co-expression patterns in neighbors? Or did they explicitly feed in that external biological knowledge about interactions?
They fed it in. And this is another place when the text-centric C2S architecture gave them a huge advantage.
How so?
They didn't have to build some complicated data fusion module to combine interaction databases with the transcriptomic data. They took existing, highly structured molecular interaction databases.
Like which ones?
Well, CellPhoneDB, which is great for known receptor-ligand pairs, the things cells use to talk to each other directly. And also BioGRID, which catalogs more general protein interactions inside and between cells.
Okay, structured databases. How do you get that into an LLM that eats text?
They converted the relevant interactions from these databases into natural language prompts.
Wait, really? You turn database entries into sentences?
Yeah. They'd filter the database first, maybe only keeping interactions involving genes actually expressed in their spatial dataset, or focusing on cell surface proteins from CellPhoneDB. Then they format these interactions as descriptive text instructions.
Can you give an example? What would that look like? So imagine the model is analyzing a specific cellular neighborhood. The input prompt would contain the cell sentences of all the cells in that neighborhood, followed by some explicit textual knowledge like known physical interactions relevant here include receptor X binds to ligand Y. Protein Z is known to inhibit kinase W. Ah, okay. So the LLM gets the raw data, the cell sentences of the neighbors, and the potential rules governing their interaction, the textual interaction network, all at the same time in the same input window.
Exactly.
And then it can use its reasoning power to figure out which of those textual rules best explains the patterns it's seeing in the cell sentences.
That's precisely the idea. And the results really supported this approach.
Yeah.
When they combined the external knowledge from both cell phone DB and BioGRID formatted as text, they saw the biggest improvement in spatial neighborhood prediction accuracy. It dramatically outperformed baseline models that only relied on the transcriptomic data itself. The LLM implicitly learned how to use that textual knowledge about molecular communication to make better guesses about physical location.
That's a really elegant way to do multimodal data integration, just using the LLM's native language ability. Okay, now let's talk about refinement. Moving from just interpreting data to actually making predictions, like predicting the effect of a drug.
Right, perturbation prediction.
You mentioned that standard supervised fine-tuning, just training on historical data, can sometimes generate outputs that look statistically okay, but aren't biologically optimal. This is where reinforcement learning, RL, came in.
Yes, exactly.
Okay.
RL was necessary to push the model beyond just mimicking the training data.
Why? What's the problem with just mimicking?
Well, supervised fine-tuning SFT models, trained purely on past observations, can easily pick up historical biases or maybe learn to predict responses that were actually suboptimal in the original experiments used for training. If our goal is for the model to generate a high-quality, biologically optimized response, maybe a gene expression profile that maximizes the potential for T cells to target a cancer cell, we need a way to actively guide its output towards that specific desired biological state.
And that guidance mechanism is RL.
That mechanism is reinforcement learning, yeah. They used a specific technique called Group Relative Policy Optimization, or GRPO.
GRPO. Sounds a bit complex. Can you simplify how they used it to make the biology better?
Sure. GRPO basically fine-tunes the model's internal policy. That's how it decides which token, or in our case, which gene name, to output next. It adjusts this policy based on a reward signal.
Okay, a reward for doing something good biologic.
Right. So for the question answering task, this IQA benchmark we talked about,
Yeah.
the reward function was directly tied to that BioBERT score. Getting a higher BioBERT score gave the model a positive reward. This guided the model away from just generating answers that sounded fluent or were statistically common in the training data, and pushed it towards answers that were semantically closer, biologically aligned, with the known, high-quality, true reference answers. It rewarded biological accuracy.
Okay, maximizing functional biological accuracy, not just making plausible sentences. And how did they use RL for the drug perturbation predictions? That seems more complex.
It is. For perturbation prediction, they cleverly used RL to optimize the model's predictions against specific biologically relevant subsets of genes rather than the whole transcriptome.
Ah, so not rewarding it for getting all 20,000 genes right, but focusing the reward.
Exactly. They defined a focus reward based on key gene programs, groups of genes known to be involved in a specific biological process they cared about.
Can you give an example of that focus?
Sure. When they were doing virtual drug screening using the L1000 Touter dataset, they defined the reward function to specifically track how well the model predicted changes in apoptosis genes.
Genes involved in programmed cell death. Makes sense for cancer therapy.
Right, because inducing apoptosis is the goal of many cancer drugs. Similarly, when they trained the model on data involving cytokine stimulation immune signaling molecules,
Yeah.
the reward was focused specifically on predicting the changes in interferon response genes. Again, focusing on the key biological pathway of interest.
I see. So this targeted optimization, driven by a reward signal calculated only on the most relevant genes, made the predictions better for actual therapeutic discovery.
Significantly better, yes. It improved both performance and the model's ability to generalize to new situations for tasks that are really relevant to finding new drugs. It basically forced the model to get the important biology right. Okay, so we've got the scaling, the language model approach, the advanced reasoning, the RL refinement, all that technical heavy lifting. It feels like it was all building towards this final, really crucial step, Could the model generate a genuinely novel, context-dependent hypothesis, something you could actually go and test in a wet lab? That was the ultimate test, exactly. And they aimed it squarely back at that core cancer challenge we started with, making those invisible cold tumors visible to the immune system.
Right. So the specific therapeutic challenge they set for the AI was this. Find a compound, a drug, that acts as an interferon conditional amplifier of antigen presentation, specifically boosting the expression of MHC class I molecules. Okay, let's quickly recap that for everyone. MHC class I, or HLA in humans, that's the complex on the cell surface that basically holds up little bits of protein from inside the cell, the antigens.
The molecular flags.
Right, the flags for the immune system, especially T cells, to inspect. If a tumor cell hides these MHCI molecules, it becomes effectively invisible, cloaked.
Correct. And interferons, or IFN, are natural immune signaling proteins that normally tell cells to make more MHCI to increase their visibility.
But the problem in many cold tumors is that the natural interferon levels are too low, not enough signal to really trigger a strong immune response.
Exactly. The signal is there, maybe, but it's too weak. So the AI was tasked with finding a drug that could effectively lower the activation threshold for that interferon signal.
Okay, so find a compound that maybe by itself doesn't do much to MHCI,
Right.
but when it's combined with those normally weak endogenous IFN signals already present in the tumor microenvironment, it would dramatically amplify the response, basically flip the visibility switch even with a weak initial signal.
That's precisely the goal. And achieving that requires very sophisticated, context-dependent reasoning, understanding how a drug works differently, depending on the presence or absence of this other signal, the interferon, which is exactly the kind of complex reasoning C2S-Scale at 27B parameters was built for.
Okay, so to find such a drug, they needed to run a virtual screen. But not just any screen, one that explicitly tested the drug under two completely different biological contexts. Tell us about the design of this dual context screen.
Yeah, this was key. They used the trained C2S-Scale model to simulate the effects of over 4,000 existing small molecule compounds. These came from a well-known library called L1000
Okay, 4,000 compounds.
And the simulation compared the predicted effect of each compound on the expression of the MHCI antigen presentation gene program. But crucially, it did this in two distinct settings.
Setting one.
Setting one was the immune context positive condition. For this, they used transcriptomic data derived from complex, real primary human tumor samples. These samples were chosen because they exhibited low but detectable levels of endogenous interferon signaling. So it mimicked a real-world subtle immune environment found in some tumors. Okay, context one, real tumors, weak IFN signal present. What was context two? Context two was the immune context neutral condition. For this, they used data from an isolated cancer cell line grown in a dish specifically, the WAGA cell line, which was known to completely lack any intrinsic IFN signaling, a clean baseline.
Got it.
So they were explicitly asking the AI, for each of these 4,000 drugs, predict its effect on MHCI genes when IFN is weakly present, and predict its effect when IFN is totally absent. They were looking for the difference, the interaction between the drug and the IFN environment. Exactly. They were hunting for compounds that showed the most dramatic context split in the predictions.
And did they find one? Which compound emerged?
C2S-Scale nominated the CK2 inhibitor silmetassertib, also known as CX-4945, as the top candidate.
Silmetassertib, okay. And why was it the top hit?
Because the model predicted a remarkably pronounced context split for this specific drug.
What was the specific prediction? How big was the split?
The prediction was really clear-cut. The model predicted a strong, robust increase in the expression of those MHCI antigen presentation genes, but only in the IFN-positive context, the primary tumor samples.
Okay. And in the other context?
In the IFN-neutral context, the WAGA cell line, the model predicted a negligible effect, basically near-zero change in MHCI genes.
Wow. So strong effect with IFN, no effect without IFN. That is exactly the signature they were searching for, the conditional amplifier.
Precisely.
Now here's the critical question for novelty. Had silmetassertib ever been linked to enhancing antigen presentation before? Was this already known?
No, and that's crucial. Silmetassertib was known as an inhibitor of the enzyme CK2, and it had been studied in cancer for other reasons. But its specific role in enhancing MHCI expression, and especially doing it in this conditional, interferon-dependent way that was completely novel. It wasn't reported anywhere in the scientific literature.
So this wasn't just finding an existing link. It was a truly AI-generated hypothesis about a new biological mechanism for this drug.
Absolutely, a de novo hypothesis.
Okay, so the AI makes this prediction. The researchers at Yale then had to take it to the bench, to the wet lab, how do they go about experimentally confirming this context-dependent effect?
Right, the validation step is critical. They tested the hypothesis in two completely distinct human neuroendocrine cancer cell models. One was derived from Merkel cell carcinoma, the other from a pulmonary neuroendocrine tumor.
And importantly, were these cell lines part of the training data?
No, absolutely not. It's critical to note these specific cell lines were entirely excluded from the model's training data. This ensures it was a rigorous, unbiased test of the prediction on unseen biological systems.
Okay. And how did they measure if the drug was successfully painting the targets on these cells? What was the metric?
They used flow cytometry, which is a standard lab technique, to measure the actual amount of MHC-I protein, specifically HLA-A, B, and C in humans, present on the surface of the individual cells.
And the readout is?
The key metric is mean fluorescence intensity, or MFI. Basically, they tag the HLA proteins with a fluorescent marker, and the MFI tells you the average brightness per cell. Higher MFI means more HLA protein, more targets on the cell surface. It's the standard objective way immunologists quantify this.
Got it. Higher MFI equals more visible to T cells. So what did the MFI readings show when they treated the cells?
They confirmed the AI's prediction absolutely perfectly.
Really?
Yeah. When they treated the cells with silmuticirtib alone, in the absence of any added interferon, the neutral setting, there was no significant change in the HLA surface MFI, just like the model predicted.
Okay, but what happened when they added the interferon?
Oh, that's where it got interesting. When they combined silmuticirtib with a low dose of either IFN beta or IFN gamma, simulating that weak signal,
Yeah.
they saw a marked, statistically highly significant increase in the MHC-I MFI on the cell surface.
Can we quantify that? How much of an increase?
Absolutely. So in the first model, the Merkel-Fell one, combining silmuticirtib at a concentration of 1,000 nanomolar with low-dose IFN beta, resulted in a 34.9% increase in the MHC-I MFI compared to IFN beta alone.
35% higher visibility. That sounds significant.
It is. And they saw a similar effect using the other major type of interferon, IFN gamma. Then, crucially, they replicated in the second, completely independent human cell model, the pulmonary one. There, combining silmuticirtib, even at a lower 100 nm concentration with IFN gamma, caused a 28.1% increase in MHC-I MFI.
Wow. Okay, those are not subtle changes. Those are substantial increases in surface antigen presentation, which you'd expect to translate directly into better immune recognition. Definitely. It's a profound functional effect. The drug behaves exactly as the AI predicted, as an interferon conditional amplifier. It lowers the response threshold, making the cells put up more MHC-I flags even when the IFN signal is weak. So the conclusion here is pretty clear. The C2S-Scale model successfully performed this complex dual-context virtual screen and uncovered a genuinely new therapeutic mechanism, something missed by standard drug screening.
Yeah, it really demonstrated the power of using these scaled-up LLMs, not just to analyze biological data, but to actually invent novel, complex therapeutic strategies based on understanding biological context. Okay, so beyond that really exciting discovery, the C2S project also brought some significant technical innovations to the table, things that could help future biological AI research. The first one you mentioned is this new metric, SCFID, single-cell freshen inception distance, right, SCFUI.
What problem was CFID actually designed to solve? Why did they need a new metric?
Well, when you have these models, especially generative ones, that create virtual cells, new cell sentences predicting a cell state, you need a reliable way to measure how realistic those generated cells are. Are they biologically plausible? Previous methods often relied on just comparing lists of genes or simple statistical measures, which often don't capture the functional integrity of the cell very well.
Okay, so CFID borrows an idea from computer vision.
FID.
Exactly. It's a direct adaptation of the freshet inception distance, or FID, which is very widely used in computer vision to judge how realistic images generated by AI models, like GANs or diffusion models, are.
Right. In vision, FID uses a pre-trained image model like Inception v3 to extract deep features from both real and generated images. Then it compares the statistical distributions of those features. How do they adapt that for single cells? You can't feed a cell sentence into an image model. No, you can't. So the core idea is the same. Compare statistical distributions of deep features. But instead of using Inception v3, which knows about cats and dogs but nothing about biology, they swapped it out. They replaced the feature extractor with an existing, well-trained single-cell foundation model, something like SCGPT, which is already trained to understand the nuances of transcriptomic data.
Ah, clever. So you use a model that already understands biology to create a meaningful representation, a sort of biological fingerprint or embedding for both the real cells and the AI-generated virtual cells.
Precisely. This specialized model embeds the transcriptomic data, whether real or generated, into a biologically meaningful latent space, a hidden mathematical space where similar cells cluster together.
And then CFID measures the distance between the distribution of real cell fingerprints and generated cell fingerprints within that meaningful biological space, using something called the Wasserstein distance.
Exactly. It quantifies the statistical distance between those two distributions in the latent space. And why this is better is crucial. Comparing raw gene lists, especially with thousands of genes, is super sensitive to noise, to outlier genes. You can get unstable metrics that don't really reflect biological reality. But by operating in this functional latent space created by SCGPT or a similar model, SDFID captures higher order variations. It's sensitive to the integrity of entire functional gene programs within the generated cells. Are the right groups of genes working together plausibly? So it gives you a much more robust, biologically aware measure of realism. It helps researchers actually trust the quality of their generative models. Okay, that sounds like a valuable tool for the field. Now let's talk limitations. Every big model has trade-offs, and a key one here seems baked into the architecture itself, this idea of causal attention,
Right.
the autoregressive nature.
Yeah, the model reads the cell sentence sequentially, from the highest expressed gene down to the lowest. What kind of biological constraint does that inherently impose?
Well, the causal attention structure means the model is fundamentally autoregressive.
Right.
When it's predicting, say, gene number K in the sequence,
Yeah.
it can only base that prediction on the context provided by genes 1 through K1, the genes that came before it in the sequence. It can't look ahead.
And since the sequence is ranked from highest expression to lowest.
Right. This structure inherently favors modeling dependencies that flow from high expression to low expression. It gets good at learning how highly expressed genes might influence moderately expressed genes, because the highly expressed ones appear earlier in the input context. Okay? Which raises the tricky question. What about real biological cause and effect that flows the other way? What if some lowly expressed transcription factor, way down the list, is actually the master switch that triggers the upregulation of a bunch of highly expressed structural genes? That's the potential blind spot. The original paper acknowledges that this causal attention limitation could, theoretically, make it harder for the model to capture a true biological causality when it flows from low-expression genes, late in the sequence, to high-expression genes, early in the sequence.
So if a key regulatory gene is buried at position 400 in the cell sentence, the model might struggle to properly account for its influence on the genes at positions 1 through 10.
It might struggle to model its true upstream influence, effectively, yes.
Yeah.
This means the model might be excellent at learning correlations, knowing that gene A and gene B often appear together in similar ranks, but may be less effective at inferring deep low to high causal relationships.
That seems like an important distinction. Correlation isn't causation.
It's the classic trade-off.
Oh.
However, the researchers argue that for their main goals, which were largely about capturing the complex correlations needed for tasks like perturbation prediction and interpretation, this constraint was manageable.
Did they have ways to mitigate it?
They suggest that the multi-cell context approach helps. By encoding multiple cell sentences in a single prompt, a low-expression gene in cell 1, late in its sequence, can still effectively influence the high-expression genes in cell 2, early in its sequence, within the same prompt. So it kind of approximates a cross-context, potentially bidirectional influence.
Okay, a partial workaround. And what about future architectural solutions? How could they address this more fundamentally?
Well, they suggest moving beyond a purely causal structure, at least for processing the single-cell context. Future models could incorporate architectural ideas like bidirectional attention, maybe by breaking the gene sequence into chunks, or by using specific layers that can look both forwards and backwards along the gene list.
Allowing the model to see the whole sequence at once.
Essentially, yeah. Or at least giving it the ability to attend information both before and after a given gene. This would help ensure that even those lowly expressed but important regulatory genes can exert their influence across the entire predicted transcriptional landscape. Okay. And finally, the universal LLM problem, hallucination. We saw C2S-Scale gives more biologically accurate answers than general LLMs, but even so, how do we ensure it's not just generating very plausible sounding but ultimately incorrect scientific narratives, especially for those open-ended interpretation tasks? Yeah, the risk of hallucination, particularly in tasks like generating cluster captions or dataset abstracts, definitely remains. It's less of an issue for the structured prediction tasks where you have clear ground truths and metrics like CFID.
But for the interpretations,
For the interpretive task, you absolutely need domain-specific safeguards. Future work will likely need to integrate more advanced fact-checking mechanisms. Maybe the LLM's output gets cross-referenced against external knowledge graphs or databases in real time to validate the biological plausibility of its claims before a researcher accepts them as insight.
Right. Adding layers of verification. Okay, so wrapping this section up, what does this all mean for the bigger picture? C2S-Scale feels like more than just one successful model. It feels like a blueprint, maybe.
I think it is. It provides a really powerful, versatile platform for accelerating discovery. By enabling the creation of these virtual cells and allowing us to simulate complex biological processes in silico,
Yeah.
it has the potential to dramatically shorten parts of the drug discovery pipeline, maybe taking phases that used to take years down to months. And the future is clearly heading towards massive multimodal integration.
So not just SCRA, sec, and text.
No. Imagine integrating epigenomic data, how genes are regulated, proteomic data, the actual proteins present, maybe spatial transcriptomic data, and clinical outcome data, all translated into different languages that a single, massive underlying LLM can understand and reason across. That's the vision.
And critically, they open sourced the code and model weights for C2S-Scale, right? That seems important for democratizing this.
Hugely important. It really lowers the barrier to entry. Individual labs around the world don't necessarily need to spend years and millions designing their own bespoke biological AI architectures anymore.
They can focus on the data.
They can focus on the data engineering, converting their specific, unique data sets into cell sentences, and then efficiently fine-tuning these large, pre-trained models like C2S-Scale for their particular research questions. It shifts the bottleneck from expensive, specialized architectural design to more accessible data manipulation and fine-tuning. #tag outro. So today we've traced this really profound journey of the C2S-Scale model. It started with what seems like a simple, elegant trick, converting that incredibly complex, high-dimensional gene expression data into text, into the cell sentence.
Okay.
Yeah, a simple idea, but with huge implications.
Yeah.
And then they scaled it up massively, training models up to 27 billion parameters on over 1 billion tokens derived from 50 million cells. Real biological language at scale.
And that scale unlocked this complex, context-dependent reasoning ability. It wasn't just pattern matching anymore.
No, it proved its worth by executing that sophisticated dual-context virtual screen. A screen specifically designed to find subtle, context-dependent therapeutic effects that normal methods would likely miss.
And that screen pinpointed silmatassertib.
It nominated silmatassertib.
Yes.
And crucially, that AI nomination was then taken into the wet lab and validated experimentally.
Showing that the drug really does act as this interferon conditional amplifier. It helps lower the threshold needed to paint targets, those MHCI antigens, onto tumor cells that were previously trying to hide from the immune system.
It really moves AI firmly into the realm of genuine scientific discovery, of hypothesis generation, not just analysis.
The success of C2S-Scale really demonstrates that the underlying architecture of large language models, developed primarily for human language, seems universally applicable. It can learn the intricate statistical language of life itself.
Yeah, the model was trained on millions of cells, essentially, to simulate life. And it worked well enough to generate a novel, validated therapeutic mechanism in the lab. This ability to accelerate discovery, potentially far beyond human speed, it's not science fiction anymore. It's here now.
Which brings us to our final thought, something for you, the listener, to mull over.
Right. Since the speed of this AI-driven virtual drug discovery is accelerating so incredibly rapidly, what ethical frameworks, what regulatory shifts do we need to put in place urgently to ensure that these powerful new therapeutic insights are translated safely, effectively, and equitably into actual patient care?
Yeah, how does society keep pace?
We're entering an era where fundamental biological breakthroughs might be generated at computational speed.
Yeah.
Our societal structures, our regulatory processes, our ethical discussions, they need to catch up fast.
Thank you for joining us on this episode of The Future of Product. I hope you heard something today that challenged your thinking or sparked a new idea. If you enjoyed this conversation, be sure to subscribe, share it with someone building something meaningful, and drop a comment. We'd love to hear from you. You can always reach out to us directly via email at thefutureofproduct@outlook.com. You can also read more insights, essays, and articles on our Substack at The Future of Product. Just click on the Substack link in the description. Remember, the future isn't something we wait for. It's something we build. Until next time, stay curious, stay inspired, and keep building the future.